Hi everyone, today we are gonna talk about important field of chemistry like natural product chemistry. This branch focuses on studying organic compounds obtained from natural sources like from plants, marine organisms, microorganisms, also even from fungi. These compounds have been the foundation of many life-saving medicines and also essential industrial materials and even daily consumer products. Yes, characterizing the natural diversity of organic products is challenging. However, nowadays advances in mass spectrometry have allowed for discovery of thousands of new compounds over the past few decades. Mass spectrometry is used in various stages of the discovery process, ranging from sample selection, screening, and fractionation to identification and replication. Also, natural products, organic compounds, as I said before, in the range of organic chemistry is typically classified into primary and secondary metabolites. Primary metabolites essential for growth and survival, like amino acids, carbohydrates, and nucleotides. And the secondary metabolites, it's non-essential but provides ecological advantages like competition, defense, and other like signaling. Yes, they are most bioactive and widely used in medicine, in the industry. They are the main focus of natural product research. There is the alkaloids. Alkaloids, nitrogen-containing organic compounds derived mainly from amino acids. Examples like morphine, quinine, nicotine. Nicotine is used as a stimulant. And what function of this? Many have pharmacological effects acting as toxin and a stimulant. The next one is terpenoid. Terpenoid derived from the 5-carbon isoprene units. Example of menthol, taxol, and carotenoids, like the precursor of vitamin A. And the functions, it's like many serve as pigments, fragrances, vitamins, and medicinal compounds. The third one is phenolic. Phenolic compounds is containing benzene rings with hydroxyl groups derived from the shikimate or polyketide pathways. Examples for phenolic is like flavonoid, like use it as like antioxidants, and tannins also occurring in tea. Also lignin. Lignin is a structural component of wood. The fourth is the polyketide. Polyketide produced by the condensation of acetyl and malonyl coenzyme A units. Examples is like erythromycin is like antibiotic, and tetracycline also is used as a antibiotic. Many act as antibiotic, anti-cancer agents, and also toxins. The last one, glycosides. Glycoside is derived from the sugar units. Examples of this streptomycin and the cardiac glycosides. Also, cellulose structural polysaccharide in plants. It serves as energy sources and structural components. Also use it as bioactive compounds.
So thank you, Balnor. Me, Arielum. I will continue my part. How natural compounds are extracted. Natural products are organic compounds produced by living organisms such as plants, microorganisms, and marine life. These compounds, like alkaloids, flavonoids, hold immense value in medicine, agriculture, and industry. However, before they can be studied or utilized, they must be extracted from their natural sources. The extraction process is a crucial and critical step in natural products chemistry, ensuring that these compounds are isolated in pure forms for further analysis. And this is the three widely used techniques for this purpose are solvent extraction, distillation, and chromatography. Each method has unique principle and applications tailored to the properties of the target compound and its sources. So solvent extraction is one of the most common and straightforward methods for isolating natural compounds. This technique relies on the principle of like dissolves like, where a solvent, typically an organic liquid like ethanol, hexane or water, is used to dissolve compounds from a natural source such as plant leaves, roots or seeds. And the process begins by grinding or crushing the raw material to increase its surface area, allowing better contact with the solvent. And the mixture is then soaked or stirred, enabling the solvent to penetrate the material and extract the desired compounds. For example, to extract caffeine from coffee beans, ethanol or water can be used as a solvent because caffeine is polar and dissolves well in these liquids. So next is a distillation. Distillation is a method used to isolate volatile natural compounds, those that easily evaporate, such as essential oils from plants or aromatic compounds from spices. This technique exploits differences in boiling points to separate compounds from their source. In steam distillation, a popular form, steam is passed through the plant material like lavender flowers or eucalyptus leaves. The heat causes volatile compounds to vaporize, and the steam carries them away. The vapor is then cooled and condensed into a liquid, which separates into water and the extracted oil due to their immiscibility. A classic example is the extraction of limonene. Limonene, steam distillation isolates limonene as an essential oil widely used in fragrances or cleaning products. So next one is chromatography. Chromatography is a powerful technique for separating and purifying natural compounds from complex mixtures, often used after initial extraction methods like solvent extraction. And it works by passing a mixture through a stationary phase while a mobile phase flows over it. Compounds in the mixture interact differently with these phases based on their polarity, size, or charge, causing them to separate as they travel at different rates. So you can see this is a schematic representation of solvent extraction, liquid-liquid distribution. And this method is widely applied in isolating natural compounds, such as essential oils from plant materials. And liquid-liquid extraction or solvent extraction involves the separation of a solid between two immiscible liquid phases. It's the aqueous phase and the organic phase. Aqueous phase, the water-based phase, usually containing polar compounds, and organic phase, the organic solvent layer, which dissolves non-polar compounds such as essential oils. And this graph shows effects of solvent to sample ratios and extraction repetition on extraction efficiency.
So let's talk about structural identification. We have four main methods. First one is ultraviolet visible spectroscopy. Is based on the absorption and ultraviolet and visible light by molecules. When light passes through a sample, electrons in the molecule absorb energy and transition from a lower energy state to a higher energy state, which means from a ground state to excited state. The specific wavelengths observed depend on the presence of conjugate double bonds and aromatic rings, which have delocalized p electrons. The more conjugation a molecule has, the longer the wavelengths of absorbed light. For example, we have beta-carotene. It's a pigment which is highly conjugated system, and it absorbs at 450 nanometers given carrots for their orange color. And UV spectroscopy is useful for conjugated natural products, flavonoids and pigments by analyzing absorption spectra, and researchers can determine the chromophoric groups and degree of conjugation in natural compounds, aiding in drug discovery, food science and environmental studies. Second method is called infrared spectroscopy. Infrared spectroscopy is based on the absorption of infrared light by molecules, which causes bond vibrations such as stretching, bending, and twisting. And each type of bond absorbs at a specific frequency, producing a unique IR spectrum. This spectrum acts as a molecular fingerprint, allowing scientists to identify functional groups in a compound. So why IR spectroscopy is useful? This type of spectroscopy is an essential technique for identifying functional groups like hydroxyl, carbonyl, carboxyl groups of compound, and also it helps to differentiate between similar compounds by analyzing their unique absorption patterns. And last one is for detecting chemical impurities and structural modifications in natural products. So next one is nuclear magnetic resonance. It also a type of spectroscopy. Nuclear magnetic resonance spectroscopy is like a magnetic based fingerprint scanner for molecules. It helps scientists to see how atoms are connected inside a compound by using a strong magnetic field. And the last one is mass spectroscopy. Mass spectroscopy determines the molecular weight and fragmentation pattern of a compound. It helps confirm molecular formulas and identify specific fragments.
Thank you, Jean-Nour. So pinocyprin is a natural flavonoid found in honey, propolis, citrus peels, and plants such as pines and the licorice. So pinocyprin is actively studied in pharmaceutical industry because of its high biological activity and potential therapeutic applications. So key properties of pinocyprin. Pinocyprin demonstrates activity against both gram-positive and gram-negative bacteria, such as E. coli. So, in article, studies indicate that it affects bacterial membranes, leading to cell lysis. So, it can be produced using biotechnological methods in E. coli bacteria through the phenylpropanoid pathways. The second is anti-inflammatory activity. Inflammation is associated with COX-1 and COX-2 enzymes, which produce inflammatory mediators. So pinocyprin suppresses inflammation by reducing the production of cytokinases. So, and inhibiting signaling pathways, and it has shown protective effects in lung inflammation models in mice, for example. So the third is antimicrobial activity. Pinocyprin has been investigated as an antibacterial agent against E. coli. So in animal experiments, 100 milligram for kilogram dose demonstrated antibacterial effects. Anti-cancer activity. So pinocyprin induces apoptosis in cancer cells while sparing healthy cells. So it has been studied in colon cancer, liver cancer, and other tumor cell lines. So it may also protect the liver from carcinogens. The fifth is antifungal activity. Pinocyprin inhibits the growth of bacterium by disrupting fungal cells membrane. And the last is neuroprotective activity. So it protects neuron from damage associated with Alzheimer and Parkinson's disease. It has also antioxidant properties, protecting mitochondria in the brain, and it improves our cerebral blood flow, reduces the impact of ischemic stroke. In conclusion, pinocyprin has strong pharmacological potential. Its safety profile makes it a promising candidate for therapeutic application.
Yes, in conclusion, natural products are valuable organic compounds. Their isolation involves advanced techniques such as solvent extraction, chromatography, and also crystallization. Yes, crystallization ensuring purity and effectiveness. Structural elucidation through mass spectrometry and X-ray crystallography allows scientists to determine their molecular properties. These compounds exhibit significant in bioactive biological activities, including like antimicrobial, anti-cancer, and anti-malarial, also near-active effects, making them essential in medicine and industry. As research advances, the discovery and the modification of natural products continue to drive biological pharmaceutical innovation and biotechnology development. And this is our references. We take this information from this science. So thank you for attention.